right, the three important pieces involved in translation include mRNA or messenger RNA. And remember, this is the piece that we discussed earlier in an earlier video that we talked about during transcription. tRNA, also produced by transcription, but in a different way. This is transfer RNA, or the firecracker-looking piece here. And then the ribosome, of course, is the large complex here. It has a large subunit and a small subunit, and it actually contains ribosomal RNA. So there's actually many more different kinds of RNA, but those are the three types that we're going to discuss. All right, let's talk about tRNA for a moment. tRNA is an interesting piece of RNA. It is, the molecule we're talking about here, it looks like a firecracker in its cartoon version, but in reality, it actually looks something more like this spaghetti-looking thing. And interestingly, the nucleotides of this tRNA bind to each other in pairs, sort of the way that DNA does, except it's just one long chain that's bound up on itself. This section down here is my anticodon, and that is represented by this here, as well as this here. And the section at the top, right here, the little wick of the firecracker where the amino acid would bind, is also here and here. And those are the active spaces of the tRNA. So here and here at the bottom, those are areas that are able to bind to other molecules. So you know that the anticodon binds to a codon, and then this piece up here is going to bind to our amino acid. So they're specific for all the different codons and all the different amino acids. The tRNAs are transcribed in the nucleus in the same way that the other RNAs are. They're specific for each codon and each amino acid. Now there are 20 different amino acids, and because the code is redundant, there are actually 64 different codons, so there are many different tRNAs. Their job is to transfer the amino acids to the ribosome to be used during protein synthesis or translation. An anticodon pairs with a complementary codon found on the mRNA. So again, this anticodon here will bind to the mRNA codon. So if there's a little three-piece codon, it would bind to this anticodon. And the base pairing rules between this third base here of the anticodon and of the codon, they're not as strict. So sometimes that third pair will not follow the typical base pairing rules that we've been talking about this whole time. Because of that, and it's really just because, you see, this is a little bit bent. Because of that fact, the code is redundant in order to avoid mistakes. And I'm actually going to show you what that means a little bit later. But this is called wobble. And because of wobble, sometimes we will have a codon that codes for one amino acid, and then another codon will be a different codon, but will code for the same amino acid. So an example of that is AAG actually codes for lysine, this codon, but so does AAA. So if I have an mRNA, and here's an A, and here's an A, and then the third nucleotide is a G, it might actually match up with an anticodon that's UUU. And that's okay because of wobble. What is a ribosome? A ribosome is the machinery that's responsible for producing the polypeptide chain that we've been talking about. And they are made up of rRNA, ribosomal RNA, and protein. That's an interesting chicken and egg question. If a ribosome is responsible for making protein and it's made of protein, then who made the first ribosome? I guess we'll just never know, but it's an interesting question. Where are the ribosomes made? They are made in the nucleolus of the nucleus. They contain two subunits, a large subunit and a small subunit. One side note, the ribosomes of prokaryotes and the ribosomes of eukaryotes are slightly different in size, but of course that's just an extra little piece of information. All right, there are 3 active sites in the ribosome, the A site, the P site, and the E site. And they have longer names, but for the purposes of this lecture, we don't really need to worry about that. So the A site is the site here where this tRNA is located, and that's the site that holds the tRNA containing the amino acid that is to be added to the growing chain of polypeptide here. And then the P site is the site that's holding the tRNA that's actually grabbing hold of the polypeptide itself. And then the E site is the exit. So a really easy way to remember this is A for amino acid, P for polypeptide, and E for exit. Let's talk about the details of translation. And the purpose again of translation is to produce a polypeptide. So the first step is called initiation. During initiation, the small subunit of the ribosome is going to bind to the start codon. The start codon found on the mRNA transcript. So that's located right here. And there's a nice blow up of this right here. The start codon is always AUG. And that means that the starting anticodon is always going to be UAC, and the tRNA is always going to be carrying the amino acid methionine, or it's abbreviated MET. So MET will always be our first amino acid, and that tRNA is going to attach to the mRNA start codon. Then the large subunit of the ribosome will actually attach here. So this is a before picture, and this is an after picture, and just take note that it does take some energy to get this process started. So that is called the transcription initiation complex, and now we are ready for step 2. Now in step 2, let me just back up a bit here so we can see the whole thing. In step 2, which is called elongation, we have several things to talk about. First of all, the tRNA here will match up with a codon right here. So another tRNA bringing a new amino acid will join, and it will join to the A site. All right, and then a peptide bond will form between the 2 amino acids that are within the ribosome. Next, the polypeptide chain will actually now be located on the tRNA that's in the A site. And then finally, this entire set here will scoot over, and that's called translocation. So this tRNA will be here now, and this tRNA will be in that spot, and all this happens not because the tRNAs necessarily move, but because the ribosome moves. So everything is still bound, the anticodon to the codon. The ribosome moves over, and that means that these guys now are in a different spot than when they started. So the tRNA that was in the P site is now in the E site, and it no longer has any amino acid attached to it. And then the tRNA that was in the A site is now in the P site. So again, the growing polypeptide is still being held in the P site of the ribosome. And this will continue over and over again until we reach the end of the mRNA molecule. And when we get to the end of the mRNA, we're now ready for termination, which is the last step in translation. Termination occurs when a stop codon is reached. So stop codon is reached in the mRNA, and it would be right here. And I know that because I see this yellow thing, this little thing that looks sort of like a little pill. This is a release factor. So a release factor binds, and this release factor binds instead of a tRNA. I just wrote release binds. Release factor. Release factor binds to the stop codon, and then the polypeptide is released. The ribosomal subunits will then dissociate, which means they let go, they detach from each other, and then we're finished. Now that we've made our transcription and translation happen, we can talk about what happens to that polypeptide at the end. We've just got a nice chain of amino acids, but that's not really a protein. A protein is a fully folded polypeptide, and sometimes proteins, you may recall, have more than one polypeptide folded up, and now they're stuck together in a complex. That protein folding is really important, and it happens spontaneously Remember that amino acids have R groups or side chains, that some are hydrophobic, some are hydrophilic, some are acidic, basic, etc. And the hydrophobic amino acids will end up in the middle of a protein, and the hydrophilic ones will end up on the outside edges. So if this is representing my polypeptide chain all folded up, the ones that are water-fearing will be in the middle, and that happens by itself, but sometimes they need a little bit of help. The chaperone protein, or chaperonin, helps the polypeptide to fold correctly. You might remember that sometimes ribosomes can be either located in the cytoplasm floating freely, or they might be located attached to the endoplasmic reticulum. I used to think as a kid that they were either attached or they weren't. But the truth is they sometimes are attached and sometimes are not. What I mean is they can move around. How do they do that? This protein that's being made here, this polypeptide, the first 3 or 4 little amino acids here are serving as a signal. That's called a signal peptide. The signal peptide is going to tell the ribosome where it should go. So this small little signal will bind to this molecule here called a signal recognition particle. The signal recognition particle will then recruit the ribosome and the mRNA and the growing protein over to the endoplasmic reticulum, and it will then dock with a pore right here on the ER, and that protein will then be extruded into the lumen of the ER, into the middle, and then it can be processed further, transported. Usually this happens with proteins that need to be produced within a lysosome or some sort of vesicle, or maybe they're going to be eventually sent to the Golgi so that they can be exported from the cell via exocytosis. Last but not least, a quick discussion of the genetic code. It's important to note that this codon chart that we are using to help us learn how translation works, we would read the mRNA and we'd look at the chart, look at each codon to determine the order or the sequence of the amino acids. This chart we could use for any organism, bacteria, jellyfish, people, dogs, cats, narwhal, anything, any animal, plant, bacteria, otherwise, any living thing on Earth uses the same code. So this is one of the reasons why we can take a gene from jellyfish that glows green under UV light and we can give it to bacteria. And that's a whole other topic, but it's interesting nonetheless that bacteria can actually transcribe and translate a jellyfish gene. You'll also notice I was talking earlier about wobble and the redundancy of the code. Redundancy means, you say it more than once. It's redundant to say that. It's redundant to say that. So you can notice here that no matter what my third base in my codon, I still get the same amino acid. So mRNA triplets or codons code for amino acids in the polypeptide chain. So just as a review, here's my double-stranded DNA, and during transcription, I produce mRNA or any other kind of RNA, and that this is actually a copy of my template strand here. So if this is my 3' to 5' template, this is my 5 to 3 mRNA, and then during translation, I will use that code to produce the appropriate amino acid sequence. You can see that there are 1, 2, 3, 4 amino acids, which means we have 4 codons. 1, 2, 3, 4. And this will eventually we'll talk about mutations and what happens when things go wrong during transcription and translation. But for now, that's the end of our series on protein synthesis.